Hey everybody, welcome back to the Soil Matters. Our hosts are AKA, so Luna and I decided that, there is Mr. Av saying, wandering around the bushes.
Wandering around in British Columbia. So, of course, I forgot that I'm joining on a little earlier. My apologies.
No worries, Av. We've got Luna, now we've got Av. I'll stick on until Leighton comes on, but Av, I don't believe you've ever met Luna. She is an absolutely amazing young woman with knowledge out the flowing out of her mind. She writes for Skunk Magazine. She's a consultant, a seed breeder, a real proponent of natural farming techniques, Korean natural farming, etc. You guys are going to have a hell of a good conversation. But Luna, tell Av a little bit more about yourself so that he knows who Luna Day is.
Hey Av, it's great to meet you.
Likewise.
So I big soil biology advocate, big in microscopy. I like working with different kind of outside the box techniques, integrating different organic acids, trying to promote phytohormones in the soil, using specific consortiums of biology to promote physiological effects inside of plants, as well as amino acid profiles, enzyme profiles, integrating polysaccharides to promote diversities of biology, as well as polyphenols, different medicinal type plants, herbs, inputs to create and encourage soil health and plant health for more nutrient dense products and better aromas in cannabis flowers.
Oh, you're on mute, Av. There you go.
Luna, that is absolutely phenomenal. I'm just so, I think in a nutshell, that is just a small little course on cannabis cultivation in a regenerative manner. So looking forward to diving into every aspect that you've just mentioned.
Yeah, I'm excited.
Yeah. So I'd like to maybe even start talking about, is your focus primarily indoor cultivation or outdoor?
So I've done it all. So, I've worked on really large scale hemp farms, doing a 700-acre hemp farm in Canada. I've done greenhouse cultivation, indoor cultivation, full sun outdoor cultivation. I've been cultivating cannabis for about 14 years.
And where would you like to jump in on the conversations around where to start in your soil biology journey?
Sure. So I made a list of talking points here. Some of them are a little high level, so I was kind of thinking of starting somewhere small and then moving up to more kind of unique concepts, like experimental concepts later on. But I was thinking maybe we could talk about soil testing, like nutrient profile testing in soil, and the accuracy and efficacy of different soil testing methods in like peat-based soils, and kind of like what you think of that, kind of the benefits, the shortfalls, the assumptions that kind of come from a lot of these soil tests, and like optimizing nutrient profiles and ratios to really provide a foundation for like beneficial biology and plant health and stuff.
Sure. So are we jumping in and talking about chemistry soil tests, or are they going to be biology soil tests, or a combination of both?
Like chemistry soil tests. So, okay. So specifically, I've been doing a lot of research into different types of soil tests. We have like our Melic-3, our saturated paste, our total digest tests and stuff like that, and how they apply to these kind of like super soil, living soil, really highly amended, built soils that we use in cannabis, and knowing that they're originally made for like a sandy loam or like a clay-based soil, and how they may or may not be super applicable to these soils that we're using in cannabis. Although they do provide good information, we may get a lot of assumptions when it comes to these tests, as well as the inability to produce accurate results, or how certain things aren't as applicable. Like from what I understand, like the cation exchange capacity in terms of using like a Malix-3 on a peat-based soil may not be super applicable when it, while we have all these different biological systems going on.
And I mean, just the notion of using a Malic-3 and the fact that we may have incredibly high pHs in many of these peat-based soils, or especially if you're using a lot of amendments, you may not be getting the true cation exchange of something with that such high organic matter as well.
Right. Absolutely. So sorry, I got so prepared and now I'm all nervous.
And so most of the folks in Canada would be using A&L labs, and they may be using a combination of a saturated paste, but some may also be using a soil carbon using a Malic-3. And then most folks in the US, I find, are using Logan Labs. Is that your experience as well?
Yeah, so I've been using Logan Labs for my Malic-3 testing and saturated paste testing. And I've gotten some results that are kind of confusing. And as I kind of dove into how these tests are made and the instruments used to collect the values, they're really based on like a lot of assumptions that kind of make them difficult to completely put like my complete faith into. I have a note here. Sorry. Jeez, where did that go?
And we can also circle back to the. Oh, you found it?
Yeah. Sorry. So specifically when it comes to like phosphorus levels, that these tests are really enabled to give an accurate representation of phosphorus levels in these high peat soils because of the organic matter, like you had mentioned, that kind of throw off your pH readings as well. What do you think about like the level of accuracy, and how much can we rely on like phosphorus level test results, like values from this Malic-3?
I feel a little stronger about a Malic-3 than I do a saturated paste, right?
Okay.
So for me, and especially if you're doing a Malic-3 in an outdoor soil, we know that as soon as we throw phosphorus in our soils, it's going to be bound by, if you have relatively high amounts of clay, it's just going to bind to the aluminum in your clay and not necessarily show up in something like a saturated paste. But a Malic-3 being a hard acid should be mimicking perhaps what fungi do to release phosphorus, right? So you will get a better representation through a Malic-3 because it's a stronger acid, an acid that's trying to mimic what fungi do to extract that phosphorus. But not every soil is going to have all of that fungi to be able to extract the amount of phosphorus that might actually be there. So if you're not a biology grower and you don't have that biology, then it might be overestimating your phosphorus availability. Would you agree with that, or is that contradictory to what you're thinking?
So from what I kind of researched, is almost that the value can be all over the place, and that we can do multiple tests on the same soil sample and get pretty widely different values from this Malic-3. So I've kind of put less of my faith into it, like less of the foundation of like how I'm building my soil and what I'm going to be amending each time. But if I see that the values are low, I'll do calculations and provide what seems like an adequate amount of phosphorus to add, or potassium, or so on. But it just seems hard to rely on something that has these really widely fluctuating values when it comes to our nutrient profiles, when our ratios between our elements need to be so correctly balanced. It makes it kind of frustrating, right? But that's good to know that you think that it's more reliable than that.
I think your experience as a grower is always going to be more important than what you see on a piece of paper.
Right.
I was talking to a farmer yesterday, just reminding them that you may send in 1 cup of soil, 250 grams of soil. They're going to use 15 grams of that soil to actually measure in the Malic 3 extraction. So you're using 15 grams that could be representing 7-10 acres of soil, right? So
right.
we have to take this with a pound of salt.
Pound of salt. Yeah.
And we really do want to make sure that you don't refute your knowledge from observing that plant and being able to say, yeah, that's a purple that I'm not familiar with that cultivar. That could be a phosphorus deficiency or something. Yeah.
Right. No soil is perfectly homogeneous, right?
Right.
So important to test all over, your field or all over your beds and stuff like that.
Yeah.
You had brought up fungi in the soil as this test result, or this testing process kind of mimics fungi and the secretions of fungi and how they kind of help break down nutrients and stuff, and how the lack of fungi kind of prevents nutrient availability and things. This is something that I've looked into a lot also. Promoting fungi in my beds is a big thing that I do, with like mulching with like a hardwood like oak, and then intentionally inoculating local fruiting bodies that I'll spread the spores. I'll manually like broadcast the spores into my bed after introducing mulches and stuff like that. But also the introduction of like lower fungi and stuff. People a lot of the time use bokashi or IMO as a way to kind of promote lower fungi, which is something that I had done and have done for a long time. Specifically bokashi, I like making different bokashi recipes and kind of playing around with introducing different polysaccharide sources to promote biology and stuff like that, instead of like glucose from molasses. And I've had a lot of good results, but I've kind of dove into like what exactly this lower fungi is quite a bit. Seeing a lot of it is very likely Zygomycetes species, Mucor, Rhizopus and stuff. And I feel like this kind of sets the stage for larger fungi with just like the secretions, the stimulants, right? These biostimulants that kind of help promote these higher fungi. What do you think about like lower fungi and their effectiveness in actually producing the secretions that make these nutrients available? Do you think that we really rely more on these higher fungi, or that these lower fungi are like sufficient?
Yeah. So where's Leighton now? No, I actually was just about to. I was just about to ask you to share a little bit more on your thoughts around that, because I'm not familiar with that concept of lower and higher fungi. But I'm a big fan of using different sources of bokashi in my mix. So sometimes using a bokashi that's on a different, on rice hulls, and one that's on coffee hulls, and using different types of fibers, wheat bran, and so on. And I add those on because I think I'm going to get different fungi and bacteria. And I just wanted to mention, you mentioned oak chips, and I think that's always interesting because oak is definitely, there are very specific fungi that kind of work on oak. And so if you are using something like oak chips, it's really nice to be able to do an IMO around an oak tree to make sure that you're getting the right microbes to really break down those oak chips. So I think that was really, it tied in so nicely.
Great. Yeah. So I like to collect actual, like mushrooms, like fruiting mushrooms, right? From like your local area, and then introduce it. So like what I did most recently is I went to an arborist who has been collecting native trees from the surrounding area, local arborist, had been like chipping them all together. So get like a real diversity of different types of woods, you know, that already have like biology, different fungi and bacteria present in it. And I feel like that's a really good way of introducing like indigenous microbes to your bed. So in terms of like lower fungi, higher fungi, and like making things available, I know I keep like asking you questions. I just know you're such a smart person, and I don't have a PhD, so I'm trying to pick your brain, even though like I'm the guest on your show.
I found the PhD in a Cracker Jack box, so
yeah, okay.
I'm all excited about this, and I already had another question. Just it's a lot about your practical experience. When you say you're introducing these, are you introducing them as a mulch, or are you breaking them down to the point where you feel comfortable incorporating them into the soil?
As a mulch. I'm using it
as a mulch.
And then like eventually they're going to get kind of like worked in and stuff as I scratch like amendments in and things like that. Yeah. But you had asked me about my experience with like these lower fungi and stuff. And yeah, I'd like to touch on that a little bit. So when I was originally introduced to adding these Bukashi and IMO and stuff like that, I was originally told that it was Actinobacteria pseudohyphae, like a bacterial pseudohyphae. Yeah. And I don't know if that's super accurate or not. At this point, I kind of think that it's probably just Mucor and Rhizopus for the most part. And there was some DNA sequencing done by an active regenerative farmer in the community that confirmed that it was like 90% Rhizopus and Mucor. And so I was kind of like, you know, for a second I was like, you know, like this kind of disappointing, you know, it's not this thing, and like maybe it's not so good, and we don't like it. And so I kind of dove into this rabbit hole of like beneficial interactions of Mucor with soil, like how Mucor can benefit soil structure, and it talks about how it can really release phosphorus levels in low phosphorus soils, which I thought was really interesting. And also it has these mycotransportation properties and these synergistic properties that can connect with mycorrhizae and create like a symbiotic relationship with mycorrhizae. So we have our mycorrhizae that, you know, extends the root surface to more nutrients, you know, their hairs, you know, their hyphae extend and kind of increase the surface area for roots, but also the Mucor can create a symbiotic relationship with that mycorrhizae, which then will myceliate the top layer where all like, you know, a lot of the nutrient cycling is happening, and where, you know, our mulches and our fallen leaf matter, plant matter and stuff to like help make it more available, which I found to be really, really interesting. But also, hi Ken, do you have something to say?
Yeah, it's actually, consider it as the super highway for nutrients moving back and forth for the electrical signals that travel through the soil. That is the World Wide Web. The true World Wide Web is the mycelium. It was one of the first plants that came onto land. It breaks down all the rocks. It is the foundation. And you were talking about the different ages of mycelium. Look at where your plant is in its life cycle of the development of our planet. That's going to be more of what you want is in that age group of plants and species around the cannabis plant for where she grew up. She didn't grow up in a redwood forest. She grew up in a very earlier transitional biology. And you have to always keep that in mind. Her greatest genetic potential is when she was born in that. And so maybe that's where you want to go to have that type of a biology existing in your soil system. And I phoned Leighton, no answer. I've texted him. So I'm not sure where Mr. Leighton Morrison is, but if he jumps on, I'll throw him up right away.
Yeah. But you know, Luna, I think what I was taking from what you were saying was that, as Ken was mentioning, yeah, we give all the credit often to mycorrhizae, right? And that's the rock star, you know, in no pun intended, but it is the rock star of the fungal community, and Jeff Lowenfels really brings us out in teaming with fungi. But as you're saying, it's so much more about, well, what had to take place to create the environment for mycorrhizae to be effective, and I think that's where we got to give credit to the roadies who are making that rock star look so good, which to me is the rhizopus and the mucor, right?
Yeah, 100%. And there's a reason why it's in every soil everywhere. It's super ubiquitous, and it's inside every fruit and vegetable. And you leave your pasta out for long enough, and it's going to start growing this lower fungi that helps break it down. Right. This is just how soil functions. So we're implementing part of just how all soils work everywhere, and like encouraging it in our soil. Although I did find an interesting study just recently talking about having too high of levels of rhizopus in your soil for cannabis cultivation specifically actually affects terpene production. So that's a really interesting piece of information as well.
And can you share a little bit more about that study in terms of why they thought there was that negative correlation?
God, I should have had this prepared. I should have brought it up earlier.
Well, no, and you can always just send us the link too. We can always revisit it, and I think Leighton's going to be kicking himself for not being here, so we'll.
Mm-hmm.
I'm so I think this is super. So, but even at your thought process around why that might be.
Immediately, the first thing that comes to my mind is competitive exclusion. When you have such a high number of one kind of biology, you're kind of filling up the opportunity for other forms of biology to come in and secrete their specific organic acid profiles, enzyme profiles and things like that. And I feel like that probably would limit the diversity when you're filling all this space with one particular kind of lower fungi.
And was a study essentially saying that there was less robustness in the terpene profile, so there weren't as many terpene, or
no, like an actual decrease in the like concentration, like the percentage of terpenes in the flower.
Okay.
Yeah.
So what type of conditions would I as a grower end up creating where I have more rhizopus fungi?
What kind of? Well, I mean, immediately I would think that you're probably maybe probably too wet would be the first thing that comes to mind. Too much not fully decomposed organic matter, like too much fresh organic matter. I keep bumping my table and shaking my camera. So I'm big on not adding a whole bunch of fresh organic matter that hasn't decomposed already. I'm really big on like earthworm castings and other like mesophilic composting processes. I'm not a huge fan of thermophilic composting. I know it's very traditional and it's very effective. I just prefer insect-based products like insect frass and earthworm castings. I feel like they're safer. They have larger diversity. They haven't gone through this thermophilic process that kind of limits the amount of diversity.
Yeah.
So I'm not big on adding tons of mulch or adding tons of fresh organic matter. I know that some people are really big on like throwing fruit directly on their beds and throwing, a lot of grain or just things like that, things that haven't been fully broken down. I'm not super big on that. Like I'll take my plant matter that I've pulled, like when I'm defoliating or lollipopping, and dry it like separately a lot of times, and then reintroduce it, and then like so that it's more brown, and then add that as my mulch, just because I don't want the biology that's there to break down fresh organic matter near the stalks of my plants, right? I don't want them like super close to the stalks of my plants. I feel like that's a great way to like have like crown rot, or just breakdown of the plant at the base of the plant, stuff like that.
I think that's a phenomenal observation. In at an energetic level, in some ways, it is sending two different messages, right? You've got a very decomposition message that's being sent by trying to break down things. Like in biodynamics, we really do have to measure our energies, and sometimes you do not want a breakdown energy at the same time as what you're trying to get a growth energy, right? And so you're trying to balance the energy. So the idea of throwing fresh mulch and trying to decompose at the same time, may not be the, and that's why it's interesting, because for me, I'm often making recommendations where if you're leaving the rootstock in a living soil bed, I actually do want to put perhaps a fungal dominant compost extract or tea or a biodynamic barrel compost where I'm trying to break down that material. But at the same time, right next to it, I'm trying to grow a new plant. And I'm just wondering am I sending mixed messages to the whole living soil system there?
Definitely. So that kind of leads me into another talking point that I wanted to get to, which is promoting conditions for specific plant growth processes, like vegetative, early flowering, mid, late ripening, stuff like that. I've been getting really into specific organic acid profiles and like organic acids that have really specific physiological effects on plants and stuff like that. A lot of the time, like when I was originally introduced to Korean natural farming, there was a lot of discussion surrounding phytohormones and extracting phytohormones through the osmotic extraction process of adding, something of high osmotic pressure like brown sugar to a plant input, letting it ferment, pull the juices out, and how you're harnessing phytohormones from that plant, the organic matter. And that was something really interesting to me. And I kind of tried to dive into that concept of can you pull phytohormones from a plant with osmotic extraction, and what is the concentration of these phytohormones, and how much concentration do we need to introduce to a plant for it to really create these physiological effects? Hey, Leighton.
Hi, sorry guys. I had a little accident today at the compost yard, so I got hosed head to foot, covered in water and liquid, and so I had to come home. And yeah, so it's been an interesting Monday, but I'm here. Good to see you again, Luna.
We sent good energy your way, brother. Believe me.
So we were just talking about attempts at harnessing phytohormones to promote specific physiological effects in cannabis plants. I had mentioned KNF originally introducing this concept to me through osmotic extraction using brown sugar and the fermented plant juice input. And I kind of dove into the efficacy of this process. Is it possible to extract phytohormones through osmotic extraction, and how much phytohormones are actually present in plant inputs, and whether or not that level of phytohormones can create a physiological response in plants. From using different fermented plant inputs, FPJs, FPEs, it's really obvious that there is like a physiological response, to me anyway, when we introduce them effectively, with like an enzyme source and a microbe source specifically. But I'm very curious, and have always been really curious on what exactly is causing that response, because we're using a whole wide profile of inputs when we're doing these things, OHN, brown rice vinegar, FPJ, so on, as well as a microbe source. And we're introducing a pretty decent amount of glucose also in like brown sugar. And what comes to mind is there's a few different possibilities, right? One is, it is phytohormones. Another is the organic acids are providing the physiological response. I think it's very possible that there could be quorum sensing that's happening, where we're triggering biological communities to interact and perform in a particular way, like change their behaviors through introducing compounds that trigger like quorum sensing in the soil. So I kind of looked into it, and it seems like the amount of phytohormones present in organic inputs is so minuscule, and also they're not water soluble. A lot of the times through like phytohormone extraction processes, people are using pretty intense solvents, ethanol, hexane, naphtha sometimes, and then concentrating them from a huge amount of organic matter into a very concentrated form when using phytohormones. So to me, that feels like that's probably not very practical, that it's the phytohormones causing it. So I kind of looked into more the profile of organic acids and the ability for organic acids to stimulate specific physiological effects, as well as quorum sensing. And for people who don't know, quorum sensing is the triggering of microbial communities to behave a certain way when in presence of enough concentration of specific compounds. So when high microbial populations come in like close proximity to each other through like a particular nutrient density, or I'm sorry, microbial density, they can build up certain levels of certain compounds that then trigger them to do a process, which is usually like biological breakdown, nitrogen fixation, biofilm fermentation, disease suppression, phytohormone production, when they get to like these levels. So I'm thinking that that's probably a big part of it, those two things. I just went ranting on forever.
No, I think you're bang on with a lot of what you're saying is I think we probably have come across many people talking about how tricontinol and is a very has strong benefits within the cannabis plant, and coming from alfalfa, and it is, and it can be incredibly powerful, but the sources of that tricontinol, the sources of the alfalfa being used, make a big difference in terms of are you actually getting the true experience of tricontinol. Otherwise, it's probably just getting the benefits of having alfalfa. And I think some of our organic acids, many organic acids are phytohormones, right? Mm-hmm. And so we do see that signaling. Glucose, for example, is a not necessarily considered a phytohormone, but it is a signal. It does strong signaling within the plant, much like citric acid does, much like acetic acid. Strong signalers, arguably phytohormones as well. But I think it's so critical how you're explaining it, that it's sometimes we're being sold snake oil.
Do you think it is possible that phytohormones are responsible for these physiological responses?
I don't think we're getting. I would say that it's our definition of a phytohormone as well, right? Because to me, glucose arguably can be considered a phytohormone because it does signal within the plant.
Right, and phytohormones are organic acids, right? Right.
In some cases too, but if it's specifically that I'm getting gibberellins from kelp because I have dehydrated kelp, I'm not sure about that. I might feel more comfortable with a kelp extract, and believing that I have gibberellins from that kelp extract and using that.
So you think that it's possible then that we can be using these kind of lower forms of extraction methods to pull phytohormones like auxins, gibberellins, cytokinins, tricontinol, all this stuff. You think that it's possible that we can be integrating them through like these kind of low tech extraction processes? Sorry,
not necessarily through a fermentation process, because to me, a fermentation process may actually break down some of those acids, mm-hmm. right? Where I would feel more comfortable with other forms of an extraction method that would not look to break down those acids to amino acids or so on.
Okay.
Yeah.
Cool.
So can I hop in here for a second? Luna, What type of reactions were you seeing when using these?
So the most obvious one to notice is like stem elongation, as well as like stem thickness, just growth rate, trigger pressure.
How about early push of 7-leaf over 5-leaf? Are you seeing any kinds of reactions like that?
Not that I really noticed. I mean, I feel like that kind of just comes from like soil health, right? Like if I've never really had much issue with like, like 5-leafing, or like 3-leafing, unless it's kind of like a photo period kind of situation. But no, I wouldn't say that I noticed necessarily any kind of like leaf count as like a reaction to these different inputs and stuff like that.
because this you guys is like, we're only on the cutting edge of understanding this. And Av, you're spot on about fermentations, but I think that there's other things that come out that perhaps we're not fully understanding the interactions between all of them. For instance, working with the Muncher group, which will be on, I think, next week, and in deep diving on all of the end results of the process of aerobic digestion, which personally I didn't think was possible, but they've proven beyond a shadow of a doubt this is legit. So we've taken the end result product out of the process, and I've divided it into a few different pieces. One is the silt, the other is the microbial community, and the third is something I don't know how to put a finger on yet. I claimed it as an enzyme. I'm thinking it's more of a hormone. There's a gentleman who will probably be on this podcast in the near future. I think he's going to pop on next week to talk about his anecdotal visual reads on what's going on with, because he did a triple side by side. He did a control, he did a bottled nutrient, and then he did Muncher juice. And the Muncher juice is showing some really interesting results. Like, it's pushing 7 leaves all over the place. So the plant has sped up its metabolic process and using this. Now, the keynote to this is to understand that cannabis went into this aerobic digestion. We used cannabis plants to determine if we could knock THC off of it through this aerobic digestion process. And as a result, we got cannabis extract out of this process. So that's why I wanted to use it on cannabis. So I sent it out to 3 different people, 2 in LA, and then Billy in Illinois, I think is where he is. But we'll verify this tomorrow. But again, it goes back to, you know, this understanding of relationships. Some of the most complex, and I think, Luna, you're on spot on with these quorum sensing, these relationships between bio and chemical interactions or reactions. It's clear beyond a shadow of a doubt that these organic acids play a real critical piece of those intricate processes that we just don't have enough information on yet. And unfortunately, science really isn't going there because there's no money to be made in it, right? Who's gonna get rich, or who's gonna, you know, invest in some of that research? It's gonna have to come out of academia and only by grad students who are curious. So I think it's gonna be a while before we really start to unlock some of those. But I think that for all reasons, yes, K&F and natural farming methodologies are showing some of these kind of hormonal reactions or organic acid reactions, plant signaling. But yeah, it's very interesting stuff.
Someone want to buy me a chromatograph? I would love to do that kind of stuff, because I'm really just curious, because I feel like once we have that piece of information, we can create specific consortiums of organic acids specifically for vegetative growth, specifically for early flower, mid flower, late flower ripening, you know, growing root stimulation, things like that. And that's kind of what I've been trying to dive into, is creating like a profile of specific organic acids that aren't necessarily phytohormones, that have these physiological responses in plants, and how we can identify local plant inputs that are rich in these profiles, to then use them for a veg stimulation, an early flower transition, and then, you know, like a bulking stage and like a ripening stage and things like that.
And have you found any work done on looking at either the exudates that are produced by the cannabis plant in those varying stages, and or the rhizome microbiome of the cannabis plant at any of these stages?
So, I mean, from what I understand, you know, the root exudate compound profile is going to alter just throughout its growth, right? Throughout its stages of development.
Probably throughout the day, right?
Yeah, totally. And even with different spectrums of light, topographical spectrums of light from the sun through particulates in the sky and so on. And I love that, that's like how the plant like communicates with the soil, right? The plant knows what it needs. It communicates to the soil what it wants. And then it sends out these compounds, the carbohydrates, amino acids, the enzymes, to then create these chemical reactions that uptake specific nutrients to then grow. And if those aren't there in the correct proportions or in the proper available forms, then the plant suffers, right? As far as like your question, like what is the like specific organic acids and profile of compounds like that the growth process? I don't think that information exists. No. That would be really cool to figure out. I would love to figure that out. I don't know how you would do that. You'd have to be ripping up the roots at different, stages of growth. And
I know that there is a study out there. I mean, it was presented at the Emerald Science conference in April. But I haven't seen it published yet, that they were starting to look at the microbiome of the cannabis plant and over different stages. But and I know that there was I recently came across some work around different exudates and just looking, not as precise as you were pointing out, but trying to look at different, what type of exudates are being produced by the plant
what'd you find?
But I haven't come across. I haven't found them yet. But
oh, okay, yeah. Have you ever like dove into the specific effects that like amino acids and amino acids? We'll just stick there. The specific effects of like amino acid profiles on growth processes.
I've always had my own little recipes. This. Even pre-cannabis, just trying to when I recommend it to organic farmers, when I would recommend a hydrolysate, it was not because of the NPK values of those hydrolysates. I mean, arguably the L-amino acids would be beneficial as a protein source, nutrient source, nitrogen source, but it was always looking at the biostimulant properties of the amino acids that I was looking for.
Definitely. Yeah, very cool. I found that doing just research or studying, you might call it, that you can introduce specific species of biology and add specific amino acids and specific organic acids, and they will interact with each other to ferment specific other organic acids, but phytohormones, right? What we call phytohormones. And I've kind of been playing with that, where adding amino acids in a specific bacteria with the ideal energy source, polysaccharides specifically, to promote that. And I don't know if it really actually did anything or not, because I don't have the proper equipment to really know. But I think that it's a really cool concept that we're not really seeing be researched and explored very much. Is intentionally triggering phytohormone production in the soil through specific consortiums of biology, amino acids, and energy sources, polysaccharides.
I mean, hey, I think we're all about citizen science. We want to encourage as much of that, and always wanting to be able to share some of that knowledge. Anything you're willing to share about your experimentation, love to hear more about it.
Yeah. I'm not proud of the specific notes, but I do have. It would take me a second. Maybe I'll look it up when we're in like a conversation.
She's as bad as me, Av. She's got like 3. Actually, she's worse than me. She has 3 monitors. Okay, I only have 2 monitors. So like that's why the head's swiveling back and forth. I love you, Luna. Thank you very much for contributing your in-depth knowledge in this, and following where it all leads.
And Luna, you are providing, hopefully when cannabis becomes federally regulated in the U.S., we're going to see a lot more research, just basic. We're talking about basic agronomic research here that we can be doing. There was a student, I believe, at the University of Michigan. He was doing his honors project, I believe, looking at jasmonic acid and seeing if he could increase trichome density and THC potency. This was an honors project that he was able to do, and I think that to me is quite interesting. Unfortunately, I don't know what the results were, but that was something that just seeing, can jasmonic acid, of course, it was. I think it was just using jasmonic acid. If that could be essentially a trigger to
increase aroma and such trichome density. I love jasmonic acid. I think it's really cool. And I feel like if we are able to harness it, when we do a lot of our like masticating through, before like we start our fermentation processes, with either lactobacillus or like an osmotic fermentation or extraction, because jasmonic acid is created when we break down cell structure, right? I'm not sure exactly how that functions, but from what I understand, the process of munching and breaking and chewing, like an animal would, a plant in the wild, triggers this plant response that creates methyljasmonate and jasmonic acid to then stimulate the plant to regrow really quickly because an animal has just eaten it, right?
Maybe it helps it produce 7 leaves.
Well, you know what? I think you're hitting on all of these different notes, but the music isn't playing yet, right? So amino acids are like cell junk food. I mean, they can just gobble this stuff up, or baby food, I should say, not junk food, because it's not junk. It's really good stuff, but they can absorb it so quick. So what is it doing? It's strengthening and increasing potency in the cytoplasm. So what is the cytoplasm? The cytoplasm is the secret to all of this shit. It's this inner biological interactions that are so far, over our head at this point in time that we can't really say. So I fed this bacteria a calcium amino acid. I fed this one not a chromium, a molybdenum amino acid digest. What's happening inside those individual cells would be radically different. And of course, the cytoplasm would then be radically different. And once released, whether through consumption by a predator, pH shift, starvation, because that's something else that nobody really talks about, is that the plant can switch exudates and starve off a whole colony very quickly to the point where, they die and they release that cytoplasm. So, again, we're getting into these crazy interactions that we really don't have all the science to back up, but we do have the citizens sciencery, and we do have observation. And in my mind, observation is 50% of all science. What happened? What was the plant reaction? That's where side by sides are just so beneficial. And I think, Luna, you're onto a whole bunch of shit that's really going to drastically change the future of agriculture, whether it's, individual plants or large scale. And
God, there's just so much that we haven't.
I know.
This is like the foundation of life, like everywhere. And since it's not economically viable and just the way that our economy is structured and stuff, it's just such a tragedy that we don't have this information. Because God, it's so fascinating, isn't it? I love it.
Where it always takes me, Luna? Amniotic fluid. It always takes me. Primordial soup, amniotic fluid. What is going on that's allowing those cell division at such an incredibly fast rate? And again, these are what you're working on is these very foundational things that are in every way the key to life.
And it's so, there's so many variables in soil and between different soils, just the composition in general, the profile of everything, there's so many factors to influence all these different effects. So it makes it really hard to like pinpoint, what is it? you know, what exactly is happening and why is it happening and what caused it? Because there's so many things. And so we can be like, you know, I added this thing and it did this because of this, but it, you know, it could be potentially so many different things. But like you said, it's all about just observing the response and knowing that it is doing something, and just continue trailblazing down that path.
And have you so you've done, I'm wondering if this muncher juice is in fact, like, to some degree, would it be similar to if I took a bunch of cannabis leaves, I just cleaned up my mums, I just did up my heavy defoliation and veg. If I went and just ran that through a mixer, a juicer, and then poured some of that back on, I was just wondering if our audience or Luna or Leighton has ever done just not fermented cannabis leaves, but just cannabis juice.
So anyone who follows my Instagram knows that I'm obsessed with putting things in a blender. I do it all the time. I've actually stopped fermenting stuff as much, because I'm trying to stay away from glucose sources and stick to mostly polysaccharides, just complex carbohydrates. And so my big thing is, and it's gotten pretty popular actually, is integrating beet juice for like the inulin content, which is great for biology. And also I figure, like what you're saying is you're masticating it, right? You're throwing it in a blender and it's just chopping it up in a whole bunch of different pieces. Yes, exactly, like that right there. So so that's a compost tea I made with beet juice and oat flour instead of glucose, and it had fantastic microbial density. It had great like bacillus chaining, or what I assume to be bacillus chaining. And then at the end of that brew, I added blended aloe vera. I threw just the whole steak, spear, whatever, frond, whatever it is, of aloe vera, chopped into pieces and threw it in a blender, blended up real good, and then strained out all the fluids, added it post brew, because I don't want any kind of fermentation processes breaking down compounds that we might find desirable. And I know that aloe vera is so rich in different profile of organic acids and amino acids and enzymes.
And you would have amazing amount of betaine in the beetroot as well.
Yeah, and yes, and nitric oxide too, which is another rabbit hole that I really try. This is a perfect transition into this next talking point that I had. So another big thing is nitric oxide and the effects that nitric oxide have on plants, and its ability to stimulate metabolic and flavonoid pathways. And I'm looking for the portion of my thing that talks about nitric oxide. Nitric oxide increases seed germination, root growth, flowering, fruit ripening, mediation of plant responses to biotic and abiotic stress, involved in plant responses to drought, salt stress, pathogen attack, mitigate the negative effects of these stressors. They also regulate gene expression, or nitric oxide regulates gene expression, reduces oxidative stress, enhances plant defense responses. So this is another compound that I feel we should add to our toolbox, right? Which I feel like no one's really talking about in organic cultivation, is nitric oxide, and something we should pay attention to. We hear a lot about salicylic acid and often sometimes jasmonic acid, although most people don't know what that is. And all these other commonly known phytohormones. There are even more phytohormones that people don't usually acknowledge. But nitric oxide, I think, is one that people should really pay attention to also, which is high in beet and red beet. So I've been planting red beet as a companion plant in my beds, as well as blending it up, straining it, watering it in just raw, or also using it in my fermentation processes. I fermented it with lactobacillus. I also added a phycocyanin extract, which we can touch on in a little bit. I think that's another really cool thing. But yeah, nitric oxide, beet juice, just blending things up in general, plant inputs that you find.
Isn't that don't humans want to consume the beet juice like runners and cyclists for the nitric oxide? Is that?
Yeah, absolutely. Yeah, so it's on like pre-workout and stuff.
Yeah.
Helps
Regulator.
boost the-
Yeah, it regulates. It's like the endocannabinoid system. It regulates so many different inner processes. And I wanted to roll it back because I think we missed on a point. There's a big difference between juicing and blending. So Luna, what I really love you to get one of those food juicers, so that what it does is it presses. You're doing a cytoplasm extraction of each and every plant cell instead of just chopping it up and making the plant matter smaller so that it can be go through the screen. The pressing process delivers some much different results, and that goes back into Av, your talk about muncher juice. And Luna, as soon as we get off this today, text me your address because I'm going to send you out a gallon of the liquid, and I'm going to send you out probably like about a quart bag of the silt. Now, the silt itself, I didn't believe the results. All the nitrogen was converted into a organic form and therefore stable, not mobile. All the other minerals, again, stable, not mobile. And so I think this muncher crew has really unlocked the most amazing use of food. And so again, it's like it's going through a biogeochemical reaction throughout this aerobic digestion that's making these organic nutrients fall out of suspension, precipitate out of suspension back to their original forms. And so another interesting anecdote, one of the three cannabis growers using this came back and said, Leighton, the roots were so white, so much different than all of the other solutions and formulations that I was using. And again, all this is going to come out in the next couple of weeks as we really dive down this rabbit hole. But man, with the work that you're doing right now, I got to get you some of this shit just so you can play with it. And further opinions on again, anecdotal, just observation of what's happening, because it falls right into the work that you're doing now.
I love it.
Yeah.
So I have a juicer, but it's got like these little teeth on it, and they spin around and they blend everything. They spin around real fast. Are you talking about like a different? You talk about a press? So
yes. So I have one.
Like a screw press, Leighton?
Yes. Yeah, it's an auger.
Yeah,
so
screw press.
It just pressurizes and pushes everything through. So you get liquid, and then you get this big cake, this big turd of plant matter, for lack of a better word, right?
Yeah. And
I had a long conversation with Kevin Jodry about this years ago, because he had a program up at Wonderland Nursery for people to come and take his fresh cannabis leaves and press them into a juice. But the problem was that they realized that the juice began to degrade very quickly. So they either had to freeze it, they couldn't refrigerate it, that didn't even work. So again, it starts to ferment because of all of the cytoplasm, this release of hormones and all of the other aminos that are coming out of it. So there's a very big difference between the auger presses versus the different types of juicers that are out there.
Okay.
And I know these auger presses are no joke. They're expensive as shit, and they're a pain in the ass to clean.
Oh, lovely. Okay.
Luna, just for anyone who, and I'm sure there's people who are following you. Was that on Instagram or Twitter or
on Instagram?
On Instagram. Just for those who are old like me and not on such things. When are you doing your application of some of these fresh juices to your cannabis plant?
So the beet, I'll use it pretty much throughout as just like a food source for biology. The aloe vera, I kind of stick to veg. I keep it in like the vegetative state. I got my soil tested recently, and my nitrate level was pretty high because I use a lot of soy amino acids and aloe. So I think I was being a little heavy-handed. My values were I think like 250 or something, and it was like the end of a cycle or something, and I was like, oh shit, okay. So a little heavy-handed there. So I've kind of cut back a bit on the soy amino acids and the aloe vera. Beet juice I'll use throughout. What else am I doing? So I'm using like coconut water. Or coconut water or coconut powder. I like using kelp and stuff.
Do you use both the powder and the water, or do you use the coconut cream, or just coconut water?
Coconut water.
Okay.
I'll try to find just like high quality coconut water and just like pour it in there. Just like kelp and stuff. But I am finding that
sorry, was that sort of in flowering?
Kelp I keep to like late flower.
Oh, sorry, the coconut water.
The coconut water, I usually do like week 3.
Week 3?
Week 4, probably.
Of flower?
Yeah.
Okay.
Yeah. You taking notes?
Yeah, got it all up here. Yeah. I can't afford all this, but it sounds great.
Yeah. So like I like to do like sprouted seed teas also. So where like I sprout like corn and blend that up. I did that just recently in my indoor project at week 3 instead of coconut. And it seemed to work pretty well. I did notice a little bit of pistil burning that happened kind of shortly after. I thought was kind of interesting. But it wasn't everywhere. It wasn't uniform throughout all the genetics. It was only a couple of them. So may have been a little heavy-handed on that one too. But
you know what else? Can we reel back for a minute? Coconut cream. There's some amazing stuff coming out on the power of that. We all know the value of coconut water, but the coconut cream is, there's a gentleman out here, Agro Coco, that I work with. He's got a number of plantations down in Mexico. And of course, they're on land. They're not in brackish water, so you don't have to worry about salt buildups. He doesn't press the core as much, so you get a better response. But he's got this tons and tons of coconut cream that people just don't understand the value of it. And so he started playing around with it with plants. And it goes back to your very work that you're doing, Luna. These again are they're total friggin' hormonal bombs, because think about what that was in the plant. I got to get you some of that shit too.
Yeah, I would think of it as the fats, right?
Yeah, the fats.
Yeah.
I've played with using coconut milk also, because we understand the introduction of like omega-3 or different fats in like fish and stuff, and fish is so commonly been used. I've been trying to stay away from fish just because I feel like I kind of think of as fish products as like fuel. It's like diesel fuel almost, or like gasoline. And when you throw it on fire, which I contribute to or which I think of biology as, it just kind of explodes, right? And I've been to a lot of farms where they have been using a lot of fish, and they have a lot of like stem rot problems. Especially if they're growing like really large cannabis plants, like these plants that get older, they stay alive for much longer. Overusing fish, they see a lot of like rot problems, as well as kelp. Sometimes people just pump their plants with fish and kelp, and then they always have these issues, and they're like, oh, I don't know why I'm losing limbs. I don't know why I'm getting these rots around the base of my stems. So I've been staying away. My point being, I've been staying away from fish and looking at different sources of fats, and coconut milk. To me, kind of clicked in my head as like a good source of fats. And now you're talking about coconut cream, and that makes a lot of sense to me too, because fats are such an important part of soil structure and biology, and like water filtration,
biofilm production.
Yeah. That's a good point too.
Builds aggregates.
Builds aggregates, yes.
All right, well, get ready, because you're going to get a whole bunch of shit coming.
Oh, you sending me stuff. Cool. I'm excited. Yeah. Yes, thank you. I'm excited.
And how is the garden looking? Sorry, Av. We worked on a little side project about 3 months ago. How did everything bounce back?
there was never any problems that happened. Oh, in the beds. So yeah, okay. So let me go over for everyone that's not us. I built a new soil for my indoor project, and I got some test results back. And this is one of the reasons why I kind of brought up test result conversations with you. And my pH was 5.0. My potassium levels were in the way upper toxic levels of potassium. My sodium levels were through the roof. And I was just like, what the fuck is, like, what the fuck happened to my soil? And on the test, the Malik 3, thing, and Logan Lab's suggestions are like, nothing will grow in the soil, not in those words, but they're like, you got to fix this soil before you plant anything in it, because nothing's going to grow in it. And I had already been growing plants in it. And
the plants were fucking beautiful, Av.
They were fucking thriving. They were thriving. They were crushing.
And she's like, could the test results be wrong? And I'm like, nah, I don't think the test results are wrong. But then we start down. What else are you doing? Why is this working? So that's why I'm asking her this question, because I'm really interested. Have you? Go ahead, finish the.
Hey Ken, can you bring up an image for me on my Instagram for the viewers?
I surely can.
So
okay, we're going to be at the top of your page. How far down is it?
So number 10, the 10th latest video posted. Scroll down just a little bit, please. That one on the left, this is living soil.
This one right here.
No, one above that. That one right there. Click that one.
That one there?
Yeah, that one there. I don't know if we can maximize that image. So these are the plants that were growing in that soil in a 5.0 pH with super high levels of sodium and over the toxic values of potassium. And not only are the plants robust, we have visible white roots growing out of the surface of the soil. And now, if you...
That's as big as I can make it.
Oh, that's cool. Can you pull up another one, please?
Yep.
So if you go up a little bit, there should be a video of the bud fermentation right there. So moon diamond at 4 weeks. So that's one of my genetics.
Right here.
Mm-hmm. When I
that one there.
So that's the same soil, same plants growing at 4 weeks. The bud development is fantastic. The buds are huge and they're frosty and they smell amazing. And I haven't really done anything except for, add some beet juice, add some aloe, add some kelp, some humic acid. But I'm mostly watering, 9 out of 10 days. I'm just doing clean water. And they're just super stoked. And the soil pH is still 5.0. and everything, nothing has changed. And so it's been really interesting because I focus a lot on biology, right? And stimulating biological processes. And when I ran the numbers for the soil that I built, it all made sense. It all balanced. I follow like Albrecht soil balancing theory to build my soils. And it all made sense, right? But when I got it tested, it was all wacky. And the calcium values were pretty low. And Leighton, when I had the conversation with Leighton, he suggested that the potassium had, and correct me if I'm wrong, but that the potassium had locked up cation exchange sites so that the calcium couldn't get in. Is that correct? That was like what you had thought?
So what I was originally thinking was that because you have such high levels of biology and you're using so much humic acid, and by default getting some fulvic acid in there, you were effectively creating buffers around the root in the rhizosphere that was protecting uptake of nutrient. But yes, to some degree, potassium at that levels is fucking toxic. And so, and you all said sodium. So the sodium was the thing I was really worried about on your cation exchange sites, because that if you have that much sodium and not enough available calcium, then yeah, your cation exchange sites would have been fucked, because they were in the saturated paste test. It was like, how is anything even growing there? So again, I think I credit you for being an intuitive grower that understands these more intricate existence or co-creations or commingling of the microbes, the nutrients, but biogeochemical interactions. So yeah, and I think too, another thing is, when that plant gets harvested, you should take a chunk of the root ball and send that to Logan and have them do the soil test from, have them extract the soil from the root ball, because I'll bet you that that's kind of what's going on, is because your other practices are allowing that force field around the rhizosphere that's allowing such incredible plant reaction in such horrific soil conditions. I have no other answers for it.
I'm not entirely sure, but I did kind of figure out the cause, because I was racking my brain of what could have possibly happened, because I used the exact same soil recipe in my soil bed. Ken, can I ask you to bring up another picture, please? So I used the exact same soil recipe. If you scroll down just a little bit, in my greenhouse soil bed, it should be right below this, right there on the right. See that one next to the frog. So that's the same soil recipe that I used in my greenhouse soil bed. The only thing that I did different was my source of earthworm castings. And it took me kind of a bit to feel like that was it. And I tested my earthworm castings that I had on hand that I used to build the other soil, and they were testing at 5.0. And I was like, oh my God, like this is it. This is what it was. And so I sent it off to Logan, the castings off to Logan Labs. I was kind of speculating that the earthworm castings had a high sodium content also, for maybe some reason. That turned out to be incorrect. So I got to figure out, I use a lot of ocean-based products in my soil building, and so I think that's probably where it came from.
What was your potassium like in those earthworm castings?
I'd have to bring it up. I could bring it up.
Sometimes earthworm castings can be way off on potassium as well.
And same with compost, for whatever reason. Compost is always high in potassium.
So that could contribute to your other cation exchange capacity.
So the value found for potassium was 1375. Yeah.
That's
and what's your calcium and magnesium? Wait till you hear this out.
Calcium was 6008, and magnesium was 1356.
Can you bounce to the saturated paste test? Because that's really the reason.
That was just the earthworm castings.
That was just the castings I was using.
So that's that's not overly safe.
Yeah, so I used the same castings when I was planting my soil bed outside, and it actually damaged all my plants, and I had to replace a whole bunch of them, and I was very upset. I was very upset because this kind of thing never happens to me, and I was so frustrated because I was like, why, And the lesson that I learned is regardless of whatever test results that you get from a supplier, you should do your own testing on every input, because it's probably not going to be indicative of the product that you've received.
Especially at the scale you're working at.
Yeah, and so the saturated paste calcium, we got 59.75, magnesium 42.05, potassium 86.39, sodium 27.18.
Right, Av? I told her, I go, whatever you're doing, Luna, you're fucking amazing. If you can get that kind of response.
So I was pissed. Because castings by nature are supposed to be neutral, right? Like the gut biomes of earthworms and stuff. You're gonna produce a neutral product, and this is what I got, and it really set back my bed.
With those numbers, I don't understand how the pH is that low. The sulfur must be like
this is why I was kind of like, what is the accuracy of these Malic 3 and paste tests? I was like, what's going on here? Like, how do these things actually work? Because none of this makes sense to me, right? So I, when I planted my greenhouse bed about a month ago, actually I think it was like 6 weeks ago, I always do like some earthworm castings at the bottom of my hole, and then I sprinkle some like mycorrhizae on top of it, and then I plant my plant directly on top of it, right? And everything immediately stopped uptaking nutrients. just stopped completely. And I added some aloe and some enzymes and like, some nice things and some beet juice, and everything just stopped growing. And I was like, it all started turning yellow, and I got purple stems, and everything's drooping and burning. There's vascular burning and all this stuff. And like this kind of thing, it never happens to me, right? And I was just furious, and it took me so long to figure out. And I was just kind of like, why would this happen? And I called Leighton, and I called a few other people, and it turns out that it was bad earthworm castings, which I guess maybe their bedding was poor, their input was poor, maybe it got too old. I don't know, but it was really big. It was a really big headache for me. But everything is now looking really good. Maybe I could post an image of the bed right now so you could see what the bed looks like now, Leighton. We could bring it up.
I can't wait to see it.
I do have.
And how much longer till you harvest that?
I'm probably going to take them to week 9. So like another month.
Oh my God, those are going to stack like crazy.
Oh yeah, definitely.
Is that a hash plant or is that a regular high tea?
It's a high tea plant. That's my moon diamond, which is a genetic that I made. It's
nice.
A DJ Shorts blueberry on kryptonite, then crossed to UK cheese, and then I put that on a cut of Diamond OG that I received that was extremely trichome dense. It almost looked like a purple punch or like a Mac 1. And I just found this fantastic phenotype of it. It's just huge buds. They're super frosty, and the smell is just cheesy and like have like berry undertones and like kind of just like gassy. And it's just, it's fantastic.
Luna,
sounds
are you growing that in another bed as well?
Everything I grow is in beds.
Oh no, sorry. Are you growing moon diamonds in another bed right now, or
like
just in a different bed?
Yeah. I had to replace some of my plants that got damaged out in my greenhouse, and so I planted some out there.
Do the plants look similar to the one in this so-called screwed up bed?
They're like a month behind the other ones.
But otherwise they look, their phenotypic expression looks the same, the smell's the same, all of that.
It's a clone. So,
but even still, because of the different soil.
I didn't really pay attention to it. I'll have to get back to you on that.
Yeah.
But I'm posting an image of my greenhouse bed right now. So you can see what the plants look like now after they've recovered and started growing. But Ken, I just posted an image on Instagram if you want to check it out or bring it up. Yeah, there it is. So that's my greenhouse bed, which got messed up originally, but I just flushed it out and did some stuff and was able to turn it around. And so I think that picture was maybe like 2 weeks ago, something like that. So now they're flowered out, and I just did some like lollipopping yesterday. So
yeah, well done.
Thank you very much. Wow. I love growing plants. I love weed.
Well, it shows, and someday I'd love to try a nugget of that creation, that moon diamond. That sounds like an amazing cross.
Aren't you in Oregon?
No, I'm in Cali, SoCal.
Oh, okay. Oh, you're in SoCal.
Yeah.
Am I going to SoCal? I need to go to SoCal. I need to visit my grandma.
Well, there you go. When you come on down, let me know.
Yeah.
Where's your grandmother?
She's in Los Angeles County.
Oh, fantastic. I'm just about an hour and a half north in Oxnard.
Okay, cool. Oh, okay. Well, I have to drive right through Oxnard to get there.
There you go. Yeah. There you go.
Cool. All right, let me bring up some other talking points. What else we got to talk about here?
I was wondering, with any of your experimentation, have you done anything with other companion planting other than just sugar beets? Anything else that others might want to try this year?
So, I like rye. I like buckwheat. I like alyssum.
Yeah.
I just threw
So
a whole bunch of
how tall do you let your rye grow?
God, until it gets in my way. So I guess it depends on the cultivar, the stretch, your lollipop, and such. But if it gets too out of control, I'll just bunch it up and get some loppers and cut it down. Wizard, thank you. And just like clean it up with like some hedge cutters, or I've seen people even use like a weed whacker and just mow it down. But I just would leave it sometimes. There was this past outdoor season that I did. I was in Southern Oregon, and I used buckwheat. I used buckwheat, and it got way too tall. And I didn't want to cut it down because it had a lot of flowers on it, and I like attracting the pollinators, and I like, just a home for beneficial insects, right? So what I did is I just rolled it. I pushed it over to the side and made like a wreath with it around the pot so that it all just like fell down and continued to cover the soil surface, but I didn't have to damage the plant and like remove any of the plant.
There's a gold bar. That's called rolling and crimping, man. That's genius because the flowers would keep growing, right?
Yeah, and so like it was almost like super cropping, almost, where you like kind of just like push it over, and I just pushed it around the pot and made like this big circle around the pot. I made it hard to top water, but we had irrigation and soaker hose and stuff, so that worked out just fine.
I've heard that chamomile releases sterols, and that those sterols are arguably some building blocks for hexanoic acid, which is one of the precursors to either maybe it's to olivetic acid, one of the precursors to THC or CBG. Have you ever experimented with chamomile as a companion plant?
No, I haven't. So so what I'm kind of hearing you say is that the root exudates have compounds present that are the building blocks for THC.
Yeah.
I always wonder if they can survive the transfer of being put into the soil and then being uptaken by another plant, and then if they can be utilized in the cellular structure of the plant.
I still think of it as a microbial food. I still think it's just all of a sudden there's something that the microbes can cheat with. It's in a greater quantity that I've got the building blocks right there. I can make hexanoic acid, or I can make
Gotcha.
Yeah, and then, I don't know if it works that way.
And so like a communicating to the soil biology. So this is kind of where that like quorum sensing that we were talking about comes in, right? Like these signaling compounds that then tell the biology to do a particular thing. So I mean, I think that that's possible, right? So like it's possible that these root exudates are then being uptaken by a cannabis plant to then build THC. Again, like who knows, but let's just do it anyway and see what happens, right?
Luna, to be honest, if you have active mycorrhizae, you are going to have those kinds of exchanges. So Av very well may be onto something as far as, in a truly healthy biological system with mycorrhizae, we know that mycorrhizae is talking to every plant in that system, and they are exchanging nutrients, exudates, and everything, including biology inside of them, endophytes. So I've got to believe that would have a definite positive effect as far as, increased THC production.
I think that it's possible for sure. I'm all about diversity of companion plants. I'm all about companion plants, living mulches, and promoting root exudates. And I've always felt like I have the most healthy, vigorous, bright green plants when there's a bunch of companion plants with it. And this is kind of like a living soil, like no-till process, really growing, where we're having a whole bunch of different types of companion plants with specific root exudate profiles that either, nitrogen fix or phosphorus fix, potassium, whatever. And then those all interact with, fungal bodies that communicate with each other, transport, use microtransportation to move specific compounds to wherever they're needed in the field or in the soil mass. And I think that concept is super invaluable, super cool, and definitely worthy of researching and implementation. I would be very curious to see the difference between a plant grown with specifically chamomile and the identical regimen and system without, and to see if it actually is doing it. And then if you could just get, maybe, and then maybe you would just get that organic acid or that compound in like a purer form, or like a consortium of these acids, and then just give it to the plant. Right. We can find these profiles of organic acids and just introduce them to the plant, because these are things that people aren't doing. Like you don't hear of everyone's selling their nitrogen or their NPK products and stuff, but no one's selling organic acid consortiums, right, to stimulate specific processes and specific compounds and secondary metabolites in plants. And I think that's where we should be going. I think that's the next place to go. Is specific arrangements of microbes.
I was always intrigued to hear Josh and Kelly explain why they use their Dragonfly Earth Medicine elixirs, and in many times it was like this. Hey, this makes sense to a pregnant mother.
Mm-hmm.
Let's put that in the soil and see what happens for essentially a
pregnant plant.
Pregnant plant.
Well, and you consider, guys, that if you have the compounds that the trichomes are being made of readily available in the soil structure around your plant, the biology doesn't have to break that out of the soil. They just take it out of that, and you're going to increase, because it's like a warehouse in a city. If your food wasn't in that warehouse to go out to the stores every day, people would go hungry. You're doing the same thing for your plant.
Well, here's the next level of that whole conversation is delivery, right? So you get a hold of these critical enzymes. I don't even know what to call them at this point, but we'll call them enzymes. And you introduce those to fulvic acid. Now it's going to be directly connected into the cell or metabolized into the cell with little or no energy. So, Luna, you're spot on about next generation to develop these critical pieces or quantum components, and then getting them into the cell on plant wise. And I think Ken, you missed the point you were trying to make, and that is, if you're saving ADP, you're allowing the plant to put its energy into other things like trichome production, like bud size, density. So that's why you're here, Luna.
Thank you. Earlier, I just want to share some information that I had collected. Earlier, we were talking about the specific consortiums of bacteria with acids to create phytohormones, and I have found some of that information I'd like to share with y'all. So for gibberellins, the combination of Bacillus subtilis, Azospirillum, and Pseudomonas fluorescens with succinic, citric, malic, and acetic acid has been shown to promote gibberellic acid in the soil.
Wait till fucking Muncher Crew gets on here. Mo starts talking about, he's been playing for 12 years with exactly what you're talking about. Consortiums of bacteria in their interactions. And that's why he's getting the results he's getting in this aerobic digestion. So again, you're totally chasing the right friggin' rabbit.
I think that this is it. I think that this is the next direction, you know, like cannabis is medicine, right? And as regenerative living soil organic grower people, you know, we understand the difference between the medicinal and therapeutic effects of an organic living soil grown plant, and not to shit on anyone, but a hydroponic one, right? And to me, it's really obvious. When you smoke it, when you taste it, the aromas are different. The profile of secondary metabolites, not just terpenes, are different. And, you know, this entourage effect is what creates medicinal effects in people, right? And so we should be promoting the full profile, the full genetic expression of the cannabis plant. And, you know, figuring out exactly what it is that contributes to these other volatile compounds that create aroma and these therapeutic effects is a crucial part of creating medicine, to me, right? So if we're really chasing medicinal value, therapeutic value, and having a superior, unique product that is a benefit to people, then these are the directions that we need to be going in, right? What is it that bring out these therapeutic effects, and how do we best harness them and implement them to create a flower that heals people and that relieves pain and grows better food too. You know, it's not just about weed, right? It's about plants, about food, regenerating soil, healing the earth, growing food that heals people and cures diseases and stuff.
And you're blazing the path, Luna. So thank you.
I just feel like I'm some weed grower on Instagram. I'd really love to blaze the path. Yeah, hopefully the universe opens up for me. Someone buy me a laboratory.
We'll work on it.
Okay.
And on one other thing on companion plants, have you ever tried potato?
Potato, not intentionally. I grew in a field that had potatoes growing in it one time.
You got to try that. There's something to it. You know, and Josh and Kelly will say the same thing. Don't know why, can't explain it, but there's some kind of interesting interaction between potato and cannabis for whatever reason. So
great.
Yeah, give it a shot. And you can get the little cute ones, or you can get big russets. It doesn't matter, just that they're in the soil near the roots. There's some kind of interaction that goes on.
Sweet. I'll try it out. Yeah, I'm all about diversity of stuff. I put a whole bunch of purple alyssum. One because of the aroma. I'm trying to encourage the floral aroma in a cannabis plant, if possible. But also it's great for beneficial insects, and it's not susceptible to things like aphids as much as some other companion plants are. But also I really just wanted a big bed full of purple flowers next to all my cannabis plants too. I think that would be really cool.
Right
on. Anyway,
so we need you to go back, or Lily suggested you go back to your presentation, because we don't want to cut you off. We only have half an hour left, and I know you are an incredible depth of knowledge. So, you know, if there's more that you want to share before we run out of time, by all means, you know, check out.
Talk about quorum sensing. Talk about nutrient tests or nutrient tests, organic acids. I want to talk a bit about phycocyanins. We talk about nitric oxide. I have another one. Horizontal gene transfer is a cool subject we could talk about in lower fungi. Let's just touch on phycocyanins real quick. So when we had talked last, you were talking about fermenting algae. You had brought up this point of fermenting algae. And so I kind of dove into algae and the profiles in algae, and found this word I hadn't been familiar with before, which is phycocyanins, which is it's responsible for like the blues. It's in a bunch of cyanobacteria. There's a lot of evidence to show that phycocyanins have a positive physiological effect on plants, specifically to stimulate plant growth, increase photosynthetic efficiency. They increase chlorophyll content in plants, increasing photosynthetic activity. can occur through a process known as non-photochemical quenching, which allows plants to regulate the amount of light energy they absorb through photosynthesis. Phycocyanins have been shown to have antioxidant properties and protect plants from damage caused by reactive oxidant species.
And these properties are why blue-green algae is so successful in our environments, because it can shade itself to collect different spectrums and survive in different types of low light or high light applications. So it only makes sense that would do the same thing in the chloroplast.
Yeah. So this is something that I had been unfamiliar with, and I feel like this is probably what we're experiencing when we're using things like kelp and seaweed extracts. But you can also just get pure phycocyanin, like 100% pure phycocyanin. They call it blue spirulina. They extract it from spirulina, and I feel like this is what people are experiencing when they make what people call super labs, where they're fermenting kelp in lactobacillus to get like this blue hue, where they have like these blue labs is what they call it, super labs. So I just got straight phycocyanin extract. And I found that phycocyanin and nitric oxide have like a really interesting interaction with each other. So phycocyanins produce nitric. Sorry, sometimes I can't talk good.
Your brain goes too quick. I get it.
So phycocyanins can stimulate nitric oxide production in plants, as well as increase the efficacy of nitrogen or nitric oxide like implementation inside of plants, like through like metabolic processes and such. So the two of them, I've paired the two of them together using beet juice and blue spirulina to stimulate plant growth as a foliar application. And I've seen some really, really cool responses. Sometimes it's hard to tell, what's doing what, but that's something I've been playing with, is the introduction of blue spirulina and beet juice as a foliar application. So this is still new. I've never heard of anyone doing this. Is something that I'm playing with. But
hey, try adding just a little fulvic acid to that mix.
Oh, okay.
Because that'll really push it into the plant cells. But I think you're onto it, which is exactly why you're supposed to be here right now.
So I added it to labs. I took lactic acid bacteria serum. I added beet juice. I added blue spirulina to it. And I started diluting that. But yeah, I'll definitely try adding some fulvic acid to it. And then using it as a foliar application. I posted it on my Instagram originally. And then I kind of like thought about it, and I was like, maybe I shouldn't put some like experimental thing that I really don't know, like what's going to happen with it. Right. Because I don't want people to like do something that I like promoted and be like, oh, like there's some phytotoxic reaction and with this specific thing that I, blah, blah, blah. And I don't want to kill anyone's plants. So, so let me put that out as a warning that this is something I just like thought of, right. That isn't necessarily widely used or potentially not without its consequences. So
Luna, it's called You're on the Cutting Edge, young lady. Just share the results. That's what we will really. You're writing the book right now, so keep writing. It's beautiful so far.
Thank you.
And just throw a warning on it. Just say, hey, this is an experiment. Please follow along.
Who knows what's going to happen?
Right.
I really don't.
And Luna, do you have a particular goal around this? What is this? Is this? Are you looking for a better quality, a different color, with a better high, a different high?
I mean, I'm always chasing aroma, right?
Aroma, okay.
Just increased metabolic and flavonoid, pathways and processes are going to produce more secondary metabolites and increase your aroma and your medicinal effects. I'm just trying to grow the best medicine possible.
And this would be arguably a flavonoid, right?
Mm-hmm.
The phycocyanin. And yeah, so it will be interesting to see if you do get different colors.
Well, because it's blue, right? It's a dye. People use it as dye.
Yeah. I mean, one of the reasons why we really do need our anthocyanins in our cannabis plant, and this is why it's so important to have both blue light but also a good full spectrum light, including green light, is because you can get a lot of these pigments in higher quantities than if you just grew under HPS, right? If you just grew under HPS, you're not going to get a lot of this other pigment production. And then of course, if your plants end up getting even more red photons, they don't have the protective pigments of the anthocyanins that can help prevent things like bleaching
Oh, cool.
and stuff, right? So I think, in my opinion, and we're still at this realm, we often are starting to attribute a better high to increased flavonoids, right? A longer high, a different type of high. And I think we're moving beyond terpenes, and now we're thinking, okay, maybe the terpenes aren't all about the high. Maybe there's something with flavonoids. So I think this type of work, might just be creating another great body experience. Yeah. A better medicine. I mean, this is why we want the color. It's why we want to eat beets and purple cabbage and blueberries and so on. We know it's got the antioxidant qualities.
Yeah, protection from oxidative stress.
Right.
And Av, I think you're spot on, about having more of the entourage, expressing more of those minor cannabinoids, flavonoids, terpenes. But there's so much more to it. But everything, like, I've been blessed to get to smoke some of the most amazing cannabis on the planet. And generally speaking, it's not chasing THC. It's high terp. It's got CBGs, CBs. It's got the whole cannabinols as well as cannabinoids. But a lot of these minor expressions that do just what you're talking about. It's a different high. It's more exhilarating. It's more thoughtful. It lasts a lot longer than those high teas. And again, Luna, this is why you're so amazing, because you're really chasing the goal of what living soil is all about. Plant expression, maximum plant genetic expression. And now you're taking it to a whole nother level with some of the work that you're doing. So yeah, man, this is, again, amazing work.
Thanks. I appreciate it. And this is great. I love talking to two big minds. It's hard to find people to talk to you about this kind of stuff.
You mean crazy minds?
Two intelligent gentlemen. It's really difficult because sometimes I'll try to talk to my friends about stuff, or just people that I know, and they're just like, I have no idea what you're talking about. I can't have these conversations with people. So like I have them with my computer or whatever. And yeah, so it's great. I really appreciate y'all having me on.
And you will be back for sure. For sure. In about 6 months, we'll have you back to give us an update on all of this that you've been working on.
Cool. Yeah, I'm always trying to come up with new stuff and trailblaze new things and go places where people aren't going. I'm not big on following other people's recipes. I'm definitely not big on doing something someone else has done.
Right. Boldly going where no one's gone before. Love it.
I'm trying to. I was a big Next Generation fan when I was a kid. So. Yeah. Okay. So what else we got here? Because we got
shout out, Mr. Toad.
Yeah, Tony. I love Tony. You're the best, Tony. I have so much respect for that guy there.
Deep respect, real deep respect.
Tony's the man.
Hey Tony, you do have access to the back end. You could always jump on for a couple of minutes.
We can talk about horizontal gene transfer.
Oh, I love this topic.
We can talk about horizontal gene transfer. Just casually end the conversation with horizontal gene transfer.
Oh, please. Tell us more. What do you got?
So horizontal gene transfer is when different organisms share genetic code with each other to adapt and overcome certain stressors or accomplish specific goals. So specifically, as it applies to soil, these goals would be like decomposition and nutrient cycling, symbiotic relationships with fungi, mycorrhizae, but also to overcome pathogenic fungi or bacteria, as well as adapt to environmental stresses. So the way that this is done is, from what I understand, is as a organism breaks down and its genetic code scatters, it can be taken on by other organisms, biology surrounding it, and use those pieces of code to rework its genetic structure to then have characteristics to overcome the list of conditions that I had just mentioned. This is best affected in high microbially dense environments like biofilms, but also in soils with a lot of biology. And this is a component of living soil that I think is often overlooked, is that in living soil conditions and thriving soil conditions, we set the stage for biology to evolve to overcome certain conditions. And so we think of evolution as survival of the fittest, as the, things get new characteristics through mutation, and then survive, right? But there's also this that comes into play, which is actually the literal bringing in of genetic code from organisms to adapt and overcome these situations, which can literally create different forms of fungi, which is why we'll never know what all the different species of fungi are, lower fungi, higher fungi, because they're constantly being almost spontaneously created, potentially on a daily basis, right? Evolution, I think, happens a lot faster than we give nature credit for. And I think that horizontal gene transfer is a big proponent of that.
Well, it's been the foundation of all evolution since the beginning of time. And I just want to touch on this. There's actually we know of 3, there may be others, but metabarcoding information has shown us that RNA is still in the soil. So metabarcoding is a way of DNA testing what's present, what's there, what's been there. And so the fact that RNA can last indefinitely in the soil means that any bacteria that comes in contact with it can take it into its outer shell, bring it into the cytoplasm, and then plug it in or excrete it, depending on what it wants to do. There's also another, when 2 particular bacteria come together and they share cytoplasm again through the outer shell. So there's a hey, I can do this. What can you do that I can't do? Okay, let's trade, right? So there's that interaction. And then lastly is the part that's really complex when you think of the consumption of a bacteria by a protozoa. There's another opportunity for this, interaction to take hold. And again, it's been the evolutionary strategy which got us to this planet to where it is that sharing, that, what does Ken call it? Sharing, caring, community, right?
Sharing, and community. Yeah. Well, you think it's like the 2 are going to university and they're only meeting for seconds, but in their life cycle, that could be almost a generation. Some 10 minutes they survive. So yeah, like it's like going to university. And hey, Tony, there's another brilliant mind.
I just wanted to come on and say thank you and respect to Luna.
Yeah. Well, you think it's like the 2 are going to university.
Sorry about that. Who's watching it? Forgot to turn it off.
Tony, what's up, man?
How you doing?
Good.
Yeah, I was stoked to see that you were going to be on here. You always have great information, so I've been watching since I got home for sure.
So you're one of the first people who really got me on this stuff. I was stumbling around with Gen Hydro, over a decade ago, and then you were one of the first people that kind of got me kicked off on all this stuff, and it just has completely changed my life and redirected everything for me. So I have tons of respect for you, and I've talked to Tony about a whole bunch of stuff throughout the years, and just knowledge bombs constantly. And so, yeah, I have a lot of respect for you, Mr. Toad.
Appreciate that. Yeah, we've spoke many times over the years, shooting
Yeah.
information off each other. It started in the PFA where that group was great in the beginning days, man. So many people learning so much new stuff about the plant, everyone coming in raw, and we all like beginners, and then all ended up with so much new information from that group, and then all of us taking it and going different directions. It's so awesome to see that.
Yeah, that's great. Yeah, still in the PFA, not super active, but yeah.
So, Av, have you ever met Tony? Or, yeah, you did on the OG's Power Hour, I think.
Tony and I have never done like a face-to-face or screen-to-screen interaction like this before. No, it's always been through text. We've just been online friends for, I don't know, for probably close to 10 years now.
Yeah, it's been, yeah. I would say so. About 10 years, yeah, for sure.
Ooh, actually, I just grew out Tony's Mary's Cherry genetic and bred with it. Super fire, chem gassy. It reminds me of like a burning rubber and like cherry. It was super awesome, and I crossed that with another cherry pie genetic, which was a cherry pie on ice cream cake. It was like a really high yielding strain. That I wanted to combine the two and kind of get this like really high yielding kind of chem dog influence. Yes, I have a whole bunch of seeds that I breed. If anyone would like to check them out, I have the listed lineages, the parent lineages of all these. These are not the strains themselves, but the parent lineages that I use to create these crosses. So if anyone's interested in that, please contact me. I have a whole bunch of seeds available. But yeah.
And Tony, I'd just like to again thank you so much for all the hard work that you've done in breeding out these types of medicines specifically for people who have needed it, and then gifting. You just give so much, so you're just a real pillar of the community. Again, deep love and respect for all.
I feel like we should all be giving more away from this plant. The plant gives so much to us. If we don't give back some of it, it's just kind of like a form of stealing. I feel like we're taking our own souls and just throwing it in the trash. This thing's our ancestor. We have so much in common with this plant that can help heal us. If we would just share the information in the plant, someone's going to do more with it than we are holding it to ourselves. Like me and Luna talking how far she's come from the beginning days. Same with myself and the rest of you guys. If we didn't have someone else give us some information to start with, we'd still be sitting there thinking that what we believed was correct. And most of the times it wasn't. You just have to open your eyes to new perspectives, and you'll find something far superior to what you already thought was true.
100%. The more we make living soil relevant and common knowledge and just open with the information to everyone, the more power that we have to improve the living soil processes and understand them, and make our products more desired, more valuable, right? So.
And better for us.
And better for us.
Right?
And now and look at how much we talked about today and really had no answers. Right?
Yeah, right?
Yeah. No questions.
Yeah.
Or answers.
I have answers to questions I haven't even heard yet.
It's true. We just talk about things, so just more and more questions. I guess that means, yeah, we're on the cutting edge, right? We're working on new things. If we don't have the
you're doing the work.
And I get skeptical. I get skeptical when people have, answers all the time too. When you ask them something and they're like, "Oh, it's this." It's like, you don't know that. So
speaking of questions, we did have a few from the chat, so we should jump on those. Does anyone recognize iodel 3 acetic acid?
Acetic acid, yeah. What about it?
Indobutanol 3, yeah. So.
Yeah, and so for example, that is a microbially. You take tryptophan and you can produce IAA through bacteria, right? Bacteria will take tryptophan, turn it into IAA, and so you can make your own. It makes its own. That's a phytohormone, right? So. Just amazing to know once again how as Luna said, these microbes are making a lot of those phytohormones as well.
So check this out: Pseudomonas putida, oxalic acid, succinic acid to promote indole-3 acetic acid.
Mm-hmm.
Yeah, sometimes we forget to give the credit to the microbes, and we talk about how I mean, like I think Luna, you mentioned it earlier. Bacillus subtilis. There's particular strains of Bacillus subtilis that actually help the plant produce the cytokinins, right? So we think it's the kelp that's doing it. Now it's actually the Bacillus subtilis that works with the roots to make the cytokinins.
Yes, and I think that distinction is very important. It's very important to be specific when we communicate these interactions, because if we lead people off the wrong trail, they're going to have correct implementation, right? Incorrect implementation. We have to understand that these things are done through biology and in the soil. It's not something.
And I think it's also important to bring up the fact that there's multiple different types of functioning bacteria. You have degraders, decomposers, solubilizers. So when these guys start interacting and pulling shit apart, it gets even crazier because they're interacting to help the work of the other one do it more efficient so it can pass along. So it's like handing the torch in one of those runs. I run for a little while, I give it to Av. He runs a little while, gives it to Tony. Tony gives it to Ken, and Ken gives it back to Luna. And we made it around the track, right? So
I dropped it a couple of times.
you were stoned, you missed it. Yeah.
Drinking again, Av? Come on, man.
He's ahead of us.
Okay, well, let's grab back to a question here, guys. And is aloe antibiotic? And yes, it is.
So
anybody want to add to that?
We think of antibiotics as like penicillin and stuff, right? Like things that we consume that I'm not even really sure how exactly antibiotics function, but a lot of the time these compounds that like have antimicrobial properties, they have those properties in like high concentrations, right? So like honey has antimicrobial properties. You leave it open and it's not going to grow like funky stuff on it, but if you mix it with water and spill it on the ground, it's going to start growing funky stuff on it. And so that's something to consider with aloe vera. And also aloe vera likes the latex. If you get the latex, like concentrations of it, like on a plant, it will damage the plant. But when it's diluted, these things don't happen. So while aloe vera has, antimicrobial properties, when it's diluted, it's not going to do that. It's not going to like kill all the bacteria in your soil. It actually promotes it. It has a polysaccharide called acetaminophen or something, which has been shown to be one of the best polysaccharides in promoting biology and bacterial formation of blooms, bacterial blooms.
And again, you can't just call it an antibiotic because there's so much more to that plant. Does it have antimicrobial properties? Yes, but there's still way beyond just an antibiotic.
Yeah, there's some.
I think that's
the amino acids that come from that plant, hormones, salicylic acid, jasmonic acid. It's a miracle plant that can help any part of your garden and yourself.
100%.
A710 ups, man.
Yeah, you know, bro.
So then we've got, so you're sort of giving your plant a smoothie.
Yeah, exactly. Pretty much. A lot of the times I'll drink the stuff that I give my plants before I give it like humic acid. I don't want to be drinking kelp and humic acid, but yeah, there's definitely times where I taste the stuff, and I see the soil as like a stomach. And we want to promote healthy gut, right? So,
well, that you mentioned inulin. I mean, that's an amazing prebiotic.
Yes, prebiotic. Yeah.
Right. So that's beautiful.
Yeah.
We should be consuming inulin as a prebiotic for ourselves.
Yeah. Every morning I have like carrot, beet, ginger, garlic, turmeric, apple juice, cucumber. I drink it. And I'm like, I should just give this to my plant. And I'm like, no, I think I'm gonna keep it. So I just kind of follow the same thinking.
Yeah, and remember, anytime you feed a plant, it's way easier for the microbes to break it down. Again, conserving energy, allowing them to work harder, faster, stronger, and better.
Yeah.
So feed your plants.
Yes. Feed your plants. Yeah, exactly.
So we're at the 2-hour mark. Does anybody want to say anything about where you're going to be? Or you got anybody have anything coming up? Luna's got the new seed drop. Anybody got anything about that?
I got some stuff coming up. I just don't have all of the hard dates. We're doing a second annual Ventura County Compost Cup out here at the Rodale House or Rodale facility. We'll also be doing a heritage seed swap.
Cool.
We'll be representing Peach Hill Soils and BioComplete Compost in September. But again, those aren't hard dates yet, and I'll definitely announce them as we move forward. But I think the big takeaway is the next 2 weeks are going to be really special, with diving down some really deep rabbit holes like today. Because I think, again, Luna, thank you so much for sharing with the community all of the hard work that you do, your dedication, your desire to boldly go where no one else has gone before, and risking it all in front of everybody. I mean, that's very commendable, and I appreciate all your hard work and the way your brain operates and where it goes. So just keep going, girl, man. Just rock and roll, brother.
Thank you.
Let's get it up.
Yeah.
I do my best. Yeah. Thank you so much. I had a blast. This is fantastic.
Well, like we said, you're going to have to come back, Luna, in 6 months and give us a harvest report. Leighton's going to send you that stuff. You're going to see some absolutely interesting results. We've got the crew from the Muncher on next Monday, and then we've got Dr. Elaine Ingham the Monday after that. I think Leighton sent her some of the Muncher stuff.
Yeah,
so she's bringing.
She's basically going to come on to discuss the test results. We all know who she is. We all understand what she's about. But this is kind of like prying open the can or the lid of Elaine Ingham's brain a little bit further into where no one's gone yet. So it should be really interesting. And again, Tony, thank you, my friend, for hopping on. It's always great to see you. And we got to drag you power hours over here at some point in the future.
Anytime, Leighton, and I respect all you guys, and thank you, Luna, for coming on here and always sharing. Appreciate that.
Thank you so much.
And Avid Ken, as always, thank you, brothers, for all the work that you're doing.
Thanks, Luna.
Thank you.
You'll have one more follower in a minute.
Fantastic. Okay, cool. Thank you so much.
If he can figure out how to use IG.
I've got a few waters. I'll figure it out.
You got grandkids. Aren't they old enough to teach Grandpa what to do?
Yeah, anyone can.
All right, guys, we're going to end it there. You guys, peace out. We'll see you on Monday with the Muncher crew.
See y'all later. Have a good night.
Thanks, Luna. Thanks, you guys.
See you.